which is a protein, disc-shaped protein who came from the outer side, bind to the centromere region, and now once there is a binding, so spindle fiber who get originated from the centriole and they attach to kinetochore protein. So remember this point, spindle fiber they attach to kinetochore protein, not directly to centromere. Okay. Second event we are talking about, that is congregation. Next one, congregation, that is whatever the genetic material, chromosome, they align at the center, they align at the equator, and such condition, there is a movement of all the chromosome at the center, at the equator. Such conditions, simply these are called as the congregation. Okay. Now, next we discuss during the condition, obviously there is a formation of the metaphase plate. All chromosomes they align at the center. That's why we are talking about these are the metaphase plate, that is plate plane of alignment of chromosome, alignment of chromosome. And in this point of view, we discuss about there is a presence of the spindle fiber. Spindle fiber basically those are made up of the microtubules, and obviously they are originated from the centriole region and they bind to kinetochore. Got it? Now, so we are talking about these types of the spindle fiber based on their exactly position. So we are talking about the continuous spindle fiber and the another one that is discontinuous spindle fiber. So first of all, we are talking about the continuous. Continuous spindle fiber, nothing but simply we are talking about they run from pole to pole. Continuous. So please write down. So we are talking about the spindle fiber who run from pole to pole. They run from pole to pole. Such type of this condition, simply it is called as the continuous one. And when you compare this point with the discontinuous, simple meaning, the spindle fiber they not reach to other pole. Discontinuous. So discontinuous nothing but that is spindle fiber not attached to or not reach to other pole. So we take the diagrammatic representation to try to understand what is the meaning of this continuous and the discontinuous. And please remember, whenever we are talking about this discontinuous type of the spindle fiber, definitely we are talking about they not attached to the pole, they are not attached to the other pole. So such type of this discontinuous spindle fiber, also we can say these are the chromosomal fiber. So such type of this discontinuous one spindle fiber, they are also called as the chromosomal fiber. So we'll see what is the exactly scenario in diagram. Just finish this part, the writing part. So we are talking about in diagram what is the meaning of the continuous spindle fiber who run from pole to pole, and what is the meaning of the discontinuous. Basically they are not reached to two pole, and such type of the spindle fiber simply is called as the chromosomal spindle fiber or chromosomal fiber. That's it. Chalo. Should I continue next part? Writing part is done. Vedant. Okay. Chalo. Yeah. So quickly we are talking about in same diagram. Okay. In same diagram we try to understand what is the meaning of the chromosomal one and chromosomal fiber, that is discontinuous spindle fiber, and what is the meaning of the continuous one. Just we discuss when we are talking about the spindle fiber basically they are showing means they run from pole to pole. So first of all we are talking about that point of view. So pole to pole running it means the spindle fiber basically they originated from the one pole and now they reach to the another pole continuously without any breaking like this. So just check, we are drawing the white color spindle fiber, and this white color spindle fiber I am giving the number 1. These are the continuous spindle fiber. Continuous spindle fiber. Why we are talking about the continuous? Because if you see they originated from the one centriole and one centriole is present toward the one pole and they reach to the another pole. So continuously without any breakage. So such type of the spindle fiber simply it is called as the continuous spindle fiber. Done?
Ma'am, please zoom out.
Yeah, sure. What you want, writing part?
No, ma'am, diagram.
Diagram na? Acha, theek hai. Continuous spindle fiber. Start from the one pole and just end with the another
Done, ma'am.
pole. Continuous hai. So these are the one spindle fiber, that is continuous spindle fiber. Now we are talking about chromosomal fiber. Chromosomal fiber also called as the discontinuous fiber. So just change your pen. कौन सा कलर ले अभी यहाँ पे? चलो, we are taking the blue color. Okay. So next point, that is chromosomal fiber or discontinuous one. So start from the one end of the microtubule, one end of the what we say, that is centriole, and they simply attach to the chromosome. Now similarly, it is not continuous like this because there is a certain gap between. So you can simply represent like this. Blue color एक attach हो गया, and now these are the showing the another side attachment. So if you clearly see the blue color dotted line, it is continuous. Definitely they arise from one end, but now what happened? They attach to the chromosome. वहां तक end हुआ. Now the another spindle fiber toward the downward direction. Chromosomal fiber or the discontinuous fiber, they get arise, and now they attach to the chromosome. Continuous nahi hai. There is a something gap. There is a something disconnection between the fiber, between the spindle fiber. So such type of the blue color, this blue color spindle fiber, simply we are talking about discontinuous spindle fiber, and such type of the discontinuous spindle fiber also called as chromosomal fiber. Also called as chromosomal fiber. Why we are talking about the chromosomal fiber? Because that spindle fiber, they attach to chromosome. Okay? Spindle fiber, they are simply attached to chromosome. That's why we are talking about the chromosomal fiber. They are not reached to second pole. Definitely they originate, they arise from the one pole, but now they're not showing the connection. Chalo. Done. If you want, if you feel it's very complicated, then you can draw a separate diagram to represent the spindle fiber, continuous fiber, and the discontinuous fiber. आप वैसे कर सकते हो, no problem. But these are the some types of the spindle fiber we already discussed. Whenever we are using the spindle fiber, definitely they originated from the centriole or they originated from the centrosome, and they mostly they perform a specific role during the, means they bind to the chromosome and simply they are pulling the chromatid material. Let's see in anaphase. So do you have any doubt? So you can draw a separate diagram to represent spindle fiber, continuous fiber, as well as discontinuous fiber. Chalo. Done this part? ठीक है. Good. Done? Chalo. Now, two important things, which is question already asked in NEET examination. Should I continue? So we are talking about some NEET point of view important question. Okay? First of all, we are talking about one drug who inhibit the metaphase. We are talking about the one drug. Name of the drug, that is colchicine. Made drug, name of the drug, that is colchicine. Basically, that colchicine, they can inhibit. So basically, these are the signs for the inhibit. One straight line and just we cross it. Inhibit ka sign hai, okay? So colchicine, basically, these are the one kind of the drug. Such drug, they can inhibit spindle fiber formation. They can inhibit spindle fiber formation. Or in other language, clearly you can say that is such colchicine, they can inhibit metaphase. Or in other language, you can see they can inhibit the mitosis process. Because now what happened? Major function, actually exactly straightforward question, very specific question. What is the function of the colchicine drug? Colchicine drug, they inhibit the spindle fiber formation, and you know very well, agar spindle formation ही नहीं हो रहा है, means spindle fiber it is not formed, it means that they're not attached to chromosome, and if you are not showing the attachment, so there is a no pulling of chromosome, no nuclear division, no karyokinesis. So colchicine, these are the one drug. We are talking about the function, that is spindle. They inhibit the spindle for fiber formation, or in other language, simply you can say they inhibit the metaphase or they inhibit the mitosis process. ठीक है? Now, basically ये जो drug है, that colchicine drug basically they obtain from the plant part, obtained from a plant part like seed and the another one that is corm, seed and the corm. So when we start the morphology chapter, that time we'll discuss what is meaning of the corm. Corm basically these are the underground stain. It's a underground stain. We'll discuss that part later when we start the morphology. So colchicine basically these are the name of the drug, and these drug obtained from the plant part. Okay, plant part, and which part? Number one seed and the another one that is corm. CORM, not N, it's M, corm. Corm basically it is example of the underground stain. Now what happened when you compare this point, means we are talking about, ये तो हमने देखा, that is a plant part. Now I am taking the example of the name of the plant. So one question, actually ये जो part है ना colchicine, this part is included in 3 to 4 chapter. It is included in chapter, basically यहाँ पे हम बात करें in cell cycle, cell division. When you talk about the transport, that time also this part is again we repeated. When you talk about the condition, we'll see the part of the mineral nutrition, that time also we'll see this part. Even in case of the transport हम ये देखेंगे, and in case of the 12th standard also we take this part. So remember this point, colchicine, these are the one drug, and from this drug they can inhibit mitosis, they can inhibit the metaphase, or in other language simply they can inhibit the spindle fiber formation. And example for the plant where we obtain such type of this drug, that is colchicum. Colchicum basically it is name of genus, and the species that is autumnale. So autumnale, colchicum, autumnale, these are the example of the plant, and basically this plant it is belonging to Liliaceae family. So again in morphology chapter ये point हम फिर से repeat करेंगे, because Liliaceae family basically these are the one type of the monocot family, मतलब under the monocot it is come Liliaceae family, Poaceae family. So under the monocot Liliaceae family to be become, we'll see this part in morphology chapter. So one important point, please remember, up to your 12th standard you have to remember what is the function of this colchicine drug, because ये बहुत बार repeat होगा. When in zoology also you will see this part in the diseases, drug part, disease and the drug part. Okay, so colchicine basically these are the one drug who inhibit the spindle fiber obtained from the one plant, that is colchicum autumnale. It is belonging to Liliaceae family, and which part? Basically we obtain this drug from the seed of colchicum or corm part of the colchicum. ठीक है? Done? चलो. Any doubt from the colchicine drug? Any doubt from the colchicine drug? चलो. Now we are talking about second part. Heram, I erased the previous slide entirely because today I want to share this white link in team. तो मैंने जो कल का पार्ट था मैंने उसको erase कर दिया. What you want, tell me. I'll explain that part again. इतना ही है, इतना सा ही पार्ट है. What you want?
Okay, ma'am, I got it. Thank you.
Got it now? ठीक है.
Yes.
Actually, I erased the previous part because फिर वो stuff होगा ना. So when I share this whiteboard link, ultimately it will create lots of stuff. So that's why I erased the previous one and start from the new one. Okay. So next part we are talking about. Next part we are talking about part that is second important question. So which mineral or the which ion which is required to maintain or to form the assembly of microtubule? So second important question. So for the assembly of microtubule, assembly means what? Formation, right? Means one microtubule it is attached to another. जैसे हमने देखा था alpha, beta, alpha, beta. So alpha bind to beta, then alpha, then beta. So they form a means polarization, means elongation of the microtubule, association of the microtubule. So simply we are talking about the assembly part. So write down the second important point. Assembly of microtubule. I'm writing just empty. Okay, you can write down full form. Assembly of the microtubule, there is requirement of some minerals, some ions, and we are taking the two example. Number one, magnesium is required, Mg2 plus. Magnesium is required, and as well as there is a requirement of the calcium. Okay, magnesium as well as the calcium. So please remember this point. ऑप्शन में सोडियम भी होगा, ऑप्शन में क्लोरीन भी होगा, in option manganese also given, potassium भी दिया होगा. So please remember only two ions or the two minerals they are required for the assembly, because magnesium and the calcium they act as a cofactor. Okay, so second important function, assembly of microtubules required magnesium and the calcium. So if you have the previous year question, so you will get the one question related to that point of view. For the assembly of the microtubule, what kind of the cofactor or what kind of the activator, or you can say ions, mineral is required. So answer magnesium, Mg2 plus, and the another one calcium. Done. Chalo, done this part. Done. Chalo. Now we are talking about the third phase, which is the shortest phase among the cell cycle, anaphase. The word ana represent up. Up means what? Means now they get separated. Okay. So next one, shortest or the smallest phase, that is anaphase. Anaphase basically these are the shortest phase because it's required very less time, and these are the very dramatic phase also. Dramatic phase in the sense we are talking, we'll discuss that part later. Okay. So before starting the anaphase, I just want to tell you what is meaning of daughter chromosome, what is meaning of sister chromosome. पहले उसके बारे में समझ लेते हैं. ठीक है. What is meaning of the daughter chromosome and what is meaning of the sister chromosome. So before that I'll start with some diagrammatic representation. ठीक है. Suppose these are the one cell. One cell. Simple we are taking. Inside the one cell, now there is a presence of the chromosome. Suppose two chromosomes are present. ठीक है. एक ही ले लेते हैं सिंप्लिसिटी के लिए. ठीक है. So one cell, now these are the condition where inside the cell only single chromosome is present. Now what happened? You know very well. This chromosome it is has the region of the centromere, right? और ये जितने भी arms से basically when how we represent this condition? This condition you can represent these are the one chromosome. Obviously, you can easily see that is P arm, Q arm. So you can say these are the one chromosome. In other language, you can say these are the two sister chromatid. Two chromatid. Why two chromatid? Because easily you can observe ये एक one thread है and now these are the another thread. So basically two threads are present. Two chromatin fibers are present. So one chromosome. In other language, you can say these are the two chromatin fibers. आप कह सकते हो these are the two chromatin fibers. or you can say these are the sister chromatid. Why we are talking about the sister? Because both the chromatid they attach by one centromere. So third name you can say, now you can call it as a, these are the sister chromatid. Sister chromatid. These are also correct. Okay? One chromosome or two chromatin fiber, or you can say that is sister chromatid. And why we are using the word sister? Because they are attached to, both sister chromatid attached to single centromere. They are attached to single centromere. So simply you can write down, ये जो blue color का part है, this part simply we are talking about, these are the part of the centromere. So single centromere is present. On the single centromere, two chromatin fiber, or you can say that is two sister chromatid, they get attached, right? And these are the certain kind of this typical structure is represent a one chromosome. One chromosome, or other language you can see two chromatin fiber, or you can say that is sister chromatid. Both तीनों की तीनों concept true है. यहाँ तक कोई doubt है? Any doubt? Clear? What is meaning of sister chromatid? When you read module, when you read NCERT book, आपको एक बहुत बार word मिलेगा. Sister chromatid, daughter chromatid. So that's why I'm explaining this part right now. Clear? Okay, Vedant. Chalo. Now I'm talking about what is the meaning of the daughter chromatid or the daughter chromosome. Suppose condition हो गया. These are the one chromosome. Now it is get separated. So now we are talking about sister chromatid. They get separate. Scenario क्या है देखो? Sister chromatid they get separated, right? And why such type of the sister chromatid they get separated? Because centromere now they get split. Split means what? Break hoga, separate hoga. Why the sister chromatid gets separated? Because centromere, such centromere simply they get split. Underline this word because anaphase में हमें यही देखना है. So normally what happens? We are talking about centromere gets split, and now sister chromatid they get separated from each other. So now what condition happens? ये है diagram, that is cell. Inside the one cell, simply we are talking about separation of the sister chromatid. So ये centromere split हुआ and sister chromatid also they get separated. So we will get this kind of the scenario. ठीक है. अब normally क्या होगा? These are the condition where what we say sister chromatid they get separate because initially they get attached, but centromere gets split. Centromere it is divided into two parts, and along with centromere, whatever the chromatid is get attached to that centromere, it can show movement towards the pole, right? So now simply we can say that sister chromatid gets separate. But once there is a separation, ये separation कब होगा? This separation will be occur during the anaphase because during the anaphase, sister chromatid they get separate. The chromatid they start moving toward the two opposite direction. But now once there is a separation of the sister chromatid, now each side of the chromosome, each side, because ये दो पार्ट होगा. आगे क्या होगा देखो, जैसे सेल एंटर करेगा into telophase, now they can form a two cell like this. So these are the one cell, and inside the one cell now there is a formation of the two nuclei. These are the cells, and now inside this condition there is a genetic material is present. So in the telophase there is a, this means common cytoplasm, but there is a formation of the two nuclei. Let's see, we see that part. But whatever this part is there, ये जो part होगा individually, suppose इसको हम थोड़ा सा divide करें from the center. I just want to represent. So whatever this part is present, this one part, now it is represent daughter chromosome. अब यहाँ पे हम chromatid word use नहीं कर रहे. Now we are talking about that is daughter chromosome. It will be become daughter chromosome because sister chromatid they get separate. Cell now entered into anaphase. Now it is go forward toward the telophase, and now that daughter chromosome they further form a daughter cell. हम ऐसे ही कहते हैं ना, there is a mother cell undergo the mitosis process and now they form a two daughter cell, right? So before the daughter cell formation, there is a formation of the daughter chromosome. तो पहले sister था, now it is become daughter. After the separation of the centromere, after the splitting of the centromere, okay. So now there is a formation of the one daughter chromosome. Similarly, in second cell, this one they can represent a second daughter chromosome. They can represent a second daughter chromosome. And now further, at the end of cell cycle or the end of the karyokinesis, such daughter chromosome further they can form a daughter cell. Further at the end they can form a daughter cell. थोड़ा सा you have to practice it, you have to revise it again and again. एक बार समझ में आ गया, so now almost up to 2 years or further, you will easily understand the concept, what is meaning of when we have to use this sister and when you have to use the daughter. Daughter means what? Two individual chromosomes, they are not attached with each other, that's why we are using the daughter. Okay. When you are using the word sister, because the chromosome or the chromatin fiber, they are attached to centromere, abhi tak attachment hai, that time we have to use the word sister. Got it? So two sister chromatid are called as the daughter chromosome in anaphase. हाँ, yes, you can say that part. But abhi, just wait. I'll see. First of all, we write down all the parts. What kind of the event will be occur in anaphase. Then we discuss the diagrammatic representation for the anaphase. तब हम आपको बताएंगे daughter chromosome. Hmm? चलो हो गया. Done. Okay, Aisha. ठीक है चलो. So first of all, we are talking about some important point about the anaphase. So we start with the information with ana. One point, these are the smallest phase or the shortest phase. Second event, basically these are the phase where centromere it will be get split. Centromere split. Due to splitting of that centromere, obviously that sister chromatid they start moving towards the opposite pole. Sister chromatid they start moving towards opposite pole where the centrioles are present, where the centrosomes are present. So simply they are start the moving. Okay. And next event we are talking about whatever the spindle fiber is present, spindle fiber as well as chromosomal fiber, spindle fiber as well as the chromosomal fiber, they get shorter. Okay, they get shorter. And the next event, there is elongation, elongation of a third and the last fiber, that is interzonal fiber. Interzonal fiber. ठीक है. So first of all, complete this three parts, which is for Anna. These are the for Anna phase, and now we are taking the diagrammatic representation. Try to understand how they are showing the movement, how it is exactly look, what is meaning of the interzone, etc. etc. चलो. Dan, should I continue? Should I continue, students? Okay, chalo. So try to understand with the help of the diagram. First of all, draw with the pencil and then you can highlight it. Okay. We are talking about anaphase. So simply we start from the plasma membrane. So these are the outermost layer of the cell. That cell simply we are talking about plasma membrane. Okay. You know very well. Basically, plasma membrane does start means centrioles are present. Two centrioles. They occupy the two opposite pole means exactly location exactly opposite with each other. They occupy the two end of the cell, and we have to draw the two centrosomes or inside the centrosomes pair of centrioles present with aster ray. So quickly we draw that structure. One end हो गया, one pole, and now these are the another pole. Inside this centrosome, now the pair of centriole is present, which is lie in a perpendicular manner, like this. And now there is a formation of the ray, that is astral ray. So as it is diagram में जो मैं मेटाफेज में draw किया था, no change with respect to centrosome. ठीक है? Now, so we just write down one sentence. There is a formation of the interzonal fiber. पहला तो इवेंट हुआ, सेंट्रोमेयर स्प्लिट हुआ. स्प्लिट मींस व्हाट? ब्रेक नहीं है, मतलब नॉन फंक्शनल है. स्प्लिट मींस व्हाट? दे डिवाइड इनटू टू. ये नॉर्मल फंक्शन है. इट इज नॉट बिकम नॉन फंक्शनल. इट इज अ वेरी नॉर्मल सिनेरियो. So split means what? Centromere they divide into two parts. ठीक है? So first of all we try to draw the part of chromosomal fiber, how it is exactly look. So पहले हम start करते हैं with spindle fiber. So ये spindle fiber से green color. We are using same pen. ठीक है? So these are the green color dotted line. We are talking about spindle fiber. And just we write down one sentence. Spindle fiber basically they get shortened. Shortened means what? Depolarization. पहले elongated थे because all the chromosome they align at the metaphase, they align at the center, they align at the equator of the cell. But now what happened? That now cell it is committed to anaphase. Anaphase में क्या होगा? Whatever the genetic material is present who get replicated during the S phase, now we have to divide into two daughter cells. So slowly, whatever the chromosome which is present at the equator, which is present at the center, they are start moving toward the opposite direction. So these are the stage where we are talking about spindle fiber, they get shortened. Okay? Now, so we have to draw the structure of daughter chromosome also. How it is exactly look? So ये हो गया blue color, that is area. For the centromere, and now we are talking about these are the chromosome. Like this. ठीक है? So now what happened? Just we discuss. हम एक सिनेरियो के बारे में बात करें. Means basically what happened? cell, whatever the chromosomes which present, now that is we simply say that is sister chromatid. But now they converted into daughter chromosome. So whatever we are talking about, all this chromosome which is present in this side. ये बहुत use the pencil, okay? These both. Basically, these are represent as a daughter chromosome. Okay, daughter chromosome. And when we are talking about what is the exactly nature or what is the DNA content which is present in case of the daughter chromosome, so now they can maintain a stage that is monad, which is similar to G1 phase. I hope you remember हमने पहले discuss किया था. In case of the G1, monad condition is present, means chromosomal fibers are present. Once it is entered into S phase, now from monad it is converted into diad condition, means DNA replication हो गया. From one copy of DNA, we can form a two copies of DNA. So now that condition that is form a diad. So entire throughout the cell, diad condition is maintained. But now the anaphase where we again achieve monad condition which is similar to G1. तो यहाँ पे monad condition maintain हुआ. Similar condition for the another one, are the cell, another part, half part. Now it is represented as a daughter chromosome, and in future daughter chromosome they converted, they form a daughter cell. So again this daughter chromosome, it maintain this stage, that is which condition, that is monad stage. Okay, so there is a one very interesting question and there is a one tricky question always asked, monad with this and another one that is dyad. तो कहाँ पे क्या stage होगा? So when we are talking about the G1, in case of the G1, basically monad condition is present. One set of DNA, right? One set of DNA because normal condition होगा. When cell it is committed to S phase, now there is a DNA replication, DNA get multiplied, DNA get double, so now it is converted into dyad condition. Cell आगे proceed करेगा into G2, again into G2, the condition still it is maintained, that is dyad condition. From G2, cell it is entered into mitosis, karyokinesis, and now we are talking about the prophase. Even in case of the prophase, dyad condition ये maintain होगा. Now from the prophase, now cell committed into metaphase. In case of the metaphase, now the dyad condition it is maintained because centromere split नहीं हुआ था. But now first time in anaphase, next phase that is during the anaphase, that dyad condition it is converted into again monad. मतलब जो जीवन में हमने जो maintain किया था, that G1 condition again be achieved during the anaphase because centromere gets split, sister chromatid they get separated, and now one side of the entire chromosome, one side, because दो pole बने, right? Two poles they get formed. One side of that whatever the genetic material is present, now that genetic material or whatever the chromosomes are present, now that chromosome simply we can say these are the daughter chromosome, and the condition for the daughter chromosome that is monad, which is exactly similar to cell which is present at the G1 phase. And same for the telophase, जैसे cell telophase में enter होगा, obviously these are the condition it is get achieved, that is monad condition. ठीक है? So question कहाँ से बनेगा? Question will be designed from anaphase. Because ऐसे क्या लगता है कि now you know very well during the cell it is once it is entered into telophase, now there is a formation of the two nuclei. So clear cut scenario कि चलो जो chromosome था now it is or whatever the DNA content is present, the DNA content is equally distributed into two cells. But this distribution it is already initiated from the anaphase because during the anaphase centromere split हुआ and chromosome which is present in a sister, जो पहले sister थे, now it is converted into daughter one. So please remember, question will be asked, question will be designed from the part of the anaphase. So during the anaphase, which condition is to get maintained? Dyad condition or the monad condition? So what will be the answer? Monad. Because centromere split hoga, there is a formation of the daughter chromosome at two sides of the cell. Done? Any doubt from this point? Ruana giving the angry smiley, means angry emoji. Her comment पे angry का दे रही है response. Done. Any doubt from Anna face? Any doubt from Anna face? Chalo, should I continue next part? Should I continue next part? No? Okay, okay, chalo. So quickly we are taking some revision part. Say unit of life. I hope you remember हमने कुछ देखा था, that is types of chromosome like metacentric, submetacentric, acrocentric, telocentric. Remember, we already discussed about the different shape of the chromosome on the position of the centromere. If centromere present at the center, then metacentric. Now towards this near to the center, that is submetacentric. Now towards the or away from the center or near to the end, that is acrocentric, and exactly at the end, that is telocentric. Okay? So quickly we are talking about that part. Yeah. So quickly we are talking about that part, that is different shape of chromosome. Different shape of chromosome. तो ये पहले हम देख चुके हैं, but quickly we'll revise this part. Different shape of chromosome. So these are the different shape of the chromosome depend upon position of centromere. Okay, position of centromere. Chalo. So quickly, first of all, we are talking about first part, metacentric chromosome shape. Metacentric, you know very well. Metacentric means what? That is chromosome basically they centromere, they basically present at the center. So such kind of this condition simply it is called as the metacentric. So when we are, first of all, we see the diagrammatic representation how it is look. एक ही क्रोमोसोम ले रहे हैं for the simplicity, ज्यादा complicated नहीं बनाएंगे. So we discuss initial stage and after splitting of centromere, what happens? Centromere present at the center, and two arms P and Q are exactly they are equal in size. So upper arm basically it is called as the small P arm. P stand for petite. Petite means small. We are next year देखेंगे in genetic structure. ठीक है? And the lower arm that is small Q arm, P arm and Q arm. ठीक है? Now simply what happened? We are talking about suppose centromere gets split, right? And now cells proceed to the anaphase. So now what kind of the condition will be occur? Centromere gets split, and now we can observe V shape of chromosome during the anaphase. Exactly V shape draw करना है, okay? So that the condition during the anaphase. So I'm giving the title. Basically, these are the conditions during the anaphase. After the centromere, once it is get split, now what happen? Obviously, we are taking such kind of the scenario. ये condition था. Suppose these are the condition during the meta. These are the condition during the meta. And after the splitting, now they can form a typical type of the structure. So what kind of this arrangement? During, if we are talking about metacentric type of the chromosome, where centromere, which is present at the middle, which is present at the center. So obviously, when that centromere gets split into two, divide this, converted into two, they form a two daughter chromosome. So obviously, whatever we obtain the shape, that shape of the chromosome, that is V shape. Which shape? We obtain V shape. You can see, obviously, they can obtain it look like a V shape. These are the first part. Chalo, done. Done. Should I continue next part? Should I continue next part? Okay, chalo. Next part we are talking about, that is submetacentric. B, second part. Submetacentric. Submetacentric means what? Exactly near to the center. And during the submetacentric condition, we obtain the shape of the chromosome. Basically, these are the L shape. Which shape? L. We'll try to understand how it is exactly look. एक काम करना, ये जो diagram है ना, this one middle part. So whatever give the title, this one. These are, we are taking the all scenario for the metaphase, and after the metaphase, the third diagram, third column, we are talking about all scenario with the anaphase. So I'm not writing again and again meta and the ana. So please consider. Second column, we are taking what kind of the chromosome look, how it is look in metaphase, and in third column, we are talking about what will be the scenario during the anaphase. So quickly, we are talking about submetacentric shape of the chromosome, how it is exactly look. So these are the position of the centromere. One R basically it is small, and the another one is basically short. Like this. Okay, so you can clearly understand, when we are talking about the metacentric, both P and QR, they are equal in length. But when we are talking about the submetacentric, upper one, anyone, mostly upper one ही होता है. PR basically these are the short, and the QR basically these are the long in length. Now we are talking about what kind of the changes will be occur during the ana. So during the ana, we can obtain L shape of chromosome. कुछ इस तरह से. Okay, so it is look like which shape? That is L shape. During the anaphase, chromosome basically it is look like a L shape. Done. Third part we are talking about acrocentric chromosome. Acrocentric, nothing but we are talking about the centromere. Basically, they are present near to the end, not exactly end, near to the end. Okay? And during the acrocentric, whatever the shape we get obtained, that shape basically J shape. J shape. Okay? So draw करके ले रहे. These are the structure of the chromosome during the metaphase, which is a highly condensed structure. Upper one basically is too short because it's near to the end. And the lower one, lower arm, quite long. Exactly. Separation during the ana, and now we can obtain a very, what we say, exactly there is a J shaped structure. One arm is too short. You can check L shape, हाँ L shape में हमने थोड़ा सा long दिखाया हुआ है, but now in this condition we are talking about the J shape. So one egg is quite short, only very some part, very few part is visible, and the another part is quite long. Please don't consider as a L shape. Basically, it is a J shape. Difference कैसे पता चलेगा? Because at the one arm basically is very small as compared to L shape and the J shape. One arm, it is basically it is a sub L shape get formed during the submetacentric. So P arm or whatever the one arm, it is quite long. But when you compare this point with J, one arm it's very short like this. And when we are talking about acrocentric, we obtain shape of the chromosome during the ana, that is J shape. Okay? And the last point, that is telocentric, which is totally absent in case of the human. Telocentric. And I hope you remember, in during the telocentric, we obtain a I shape, shape of the chromosome I shape, and such type of the telocentric condition is totally absent in case of human. Okay? So we are taking the diagrammatic representation, that is centromere present exactly at the end, like this, the centromere end, and now there is a presence of only one arm, like this. Zoom out.
Ma'am, is it I shape?
हाँ, I shape. I. Hmm?
Okay, ma'am.
Hmm. So draw only one arm because centromere present exactly at the end. Telo and telomere means what? Exactly at the end. So draw only one arm. Okay? And when it gets separate, it look like a eye. Like this. Hmm? तो ये बना हमारा final shape, that is eye shape. Done? So quickly check, these are the exactly anaphasic condition: metacentric, submetacentric, acrocentric, and the telocentric. When if chromosome is metacentric, means centromere present at the middle, that time we can obtain a V shape during the anaphase. When if submetacentric chromosome is present during the anaphase, we can obtain the L shape. If acrocentric, that time we can obtain a J shape. And when there is a telocentric, which condition is totally absent in case of the human, that time we can obtain a I shape. Done? Clear this point? Any doubt? Any doubt? Bhakti. Okay. Chalo. Done? Hmm. So now we are talking about one important question. Do you have a module in front of you? So please take a photo. I want one question that is related to anaphase promoting complex. Starting की होगी देखो. Anaphase promoting complex APC. In question clearly mentioned that anaphase promoting complex or APC. I think that is a preemptive question. Just check out. No. I think 2016 नीट का एक बार देख लो. If I'm not wrong. APC के related कुछ तो question है. Anaphase promoting complex. So we are taking one question which is related to anaphase. During the anaphase, basically there is one complex is present. That complex simply we are talking about anaphase promoting complex APC. Anaphase promoting complex, and such complex simply we are talking about APC. Check the 2016 or 2012. APC के related see clearly दिया होगा वहाँ पे anaphase promoting complex APC. Just check out quickly. यार मुझे क्वेश्चन याद रहते हैं एंड सम टाइम आई रिमेम्बर ईयर आल्सो बट नॉट ऑल टाइम. एक मिनट. थैंक्स चिराग. लेट मी चेक. Check the second page. Check the second page. यहाँ पे नहीं है. Check 2016 question. नीट 2016 एक बार चेक करो. Yeah, you can see the question number 11. 2013 AIMS question. Which of the following ions it is necessary for assembly of microtubules? Check the question number 13. Not 13, sorry. Question number 11. You can check question number 11 and tell me the answer quickly. Which of the following ions it is necessary for the assembly of microtubules? In option, sodium, potassium, calcium, chlorine, calcium, magnesium, and the sodium, calcium. Yes, true. Answer will be calcium and the magnesium, because just we discussed for the assembly, for the formation, for the elongation of the microtubules, cofactors are required, ions are required, or you can say minerals are required. That is calcium and the another one magnesium. Okay. Okay. Chirag. Question number 20. Just quickly check. Question number 20. सेकेंड इमेज चिराग ने शेयर किया हुआ है, वहां पे देखना प्लीज. नीट 2017 क्वेश्चन. नीट 2017 क्वेश्चन. ठीक है. So that is simple straightforward question. APC. If APC is defective, then what happens? So we try to understand. APC it stands for anaphase promoting complex. So obviously नाम से पता चल रहा है, these are the one complex, these are the one group of the protein who responsible for its promote the anaphase, its handle the anaphase, or they can control the anaphase, because सब कुछ अच्छे से हो, everything should be normal, cell division successfully get proceed. So anaphase promoting complex, these are the one complex who activate during the anaphase, and such complex it is responsible for equal segregation. Segregation means separation. Okay, equal segregation, or you can say that is equal separation of chromosome. Equal segregation or equal separation of chromosome. So these are the exactly major function of anaphase promoting complex. So straightforward question है. In question number 20 you can check out. Anaphase promoting complex APC is a protein degradation machinery necessary for the proper mitosis in animal cell. Yes, it is true. If APC is defective, मान लो कि defective हुआ, so what happen with respect to, means what kind of the changes will be occur? So obviously first option, Chromosome will be fragmented. No, chromosome it is not fragmented. Second option देखो, chromosome will be not separated. Yes, B option it's true because if anaphase promoting complex, if epicet is become defective, अगर कुछ गड़बड़ हुआ, so there is a no proper separation of the chromosome because anaphase promoting complex it is play very important role for the equal segregation of the chromosome. So correct answer B. Done? Okay, chalo. Yeah. So any doubt from the anaphase? Any doubt from anaphase? अरे हाँ इंटरजोनल फाइबर ओके 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 गॉट इट अच्छा याद दिला दिया तो वी आर टॉकिंग अबाउट दैट इज इंटरजोनल फाइबर सेम डायग्राम में हम बात करें ओके so interzonal fiber basically it is specifically formed during the anaphase. So what is meaning of the interzonal fiber? Now there is some fiber which is formed between two daughter chromosomes like this dotted line okay like this. So these are the white color the fiber which is get formed between two daughter chromosomes. These are called as the interzonal fiber. These are called as the interzonal fiber. And what is the function of the interzonal fiber? Interzonal fiber basically they push करेंगे, not pulling. Spindle fiber, chromosomal fiber, they pull the chromatid. They pull the chromatid to opposite pole. Pulling phenomena आएगा. Now here we are talking about interzonal fiber. They form between two daughter chromosome and simply they are responsible for pushing. So basically they push daughter chromatid towards opposite pole. Okay, towards opposite pole. So please underline push कर रहे because now where exactly you can say spindle fiber they get shortened because they are showing the pulling process. If you want you can write down here they are showing the pulling process. Pulling of chromatid. Who? Spindle fiber or chromosomal fiber. Okay, spindle fiber they are showing the simple process that is pulling. But when we are talking about the interzonal fiber they are pushing because obviously spindle fiber अपनी ताकत लगा रहा है अपना जोर लगा रहा है and they are showing they are simply pulling the chromatid toward the two opposite pole as well as the interzonal fiber they get formed during the anaphase. They bind exactly at the center and they push two daughter chromatid toward the opposite pole toward the opposite direction. Got it? चलो चलो should I continue next part? These are the all scenario with respect to anaphase. Now cell proceeding toward the last phase of karyokinesis, last phase of nuclear division that is telophase. Write down the title. We are talking about the telo and telophase represent end of karyokinesis. So write down last one, last phase, telophase. Okay, and this telophase, these are the phase. It is represent end of karyokinesis. These are the end of karyokinesis. Chalo, done. So first of all, we are talking about what kind of this event will be occur during the telophase, and then we'll see the diagrammatic representation. Should I continue? Okay, Samruddhi. Chalo. Yeah. So first of all, we are talking about first part. Now, telophase, basically these are the end of the karyokinesis. It means that these are the stage where chromosome, it will be again start decondensation. Exactly opposite hoga prophase ka. Whatever the scenario we already discussed in case of the prophase, because during the prophase we already discussed condensation of the chromatin material, nuclear membrane they get disappear, nucleolus, endoplasmic reticulum, Golgi complex, all these organelles they get disappear. Exactly reverse scenario will be happen during the telophase, and now we are talking about the first stage. Chromosome slowly they are showing decondensation, and now they convert it into chromatin fiber. ठीक है चलो. So write down the first part we are talking about. That is chromosome decondensation. Okay. Chromosome decondensation, it will be begin, and now slowly that chromosome it is converted into again chromatin fiber. And obviously whenever we are talking about the chromatin fiber means we are talking about chromosome it is not present in a condensed structure. Now they simply convert it into thread. They convert it into thread. They form a thread. Okay. Yeah. So slowly, one event. Please underline the word decondensation. ये हो गया first event. Now next event we are talking about sister chromatid. Now they reach towards their respective opposite pole. So now we are talking about that is can we are using the daughter. That's better. So we are talking about that is daughter chromatid. They reach to their respective pole, exactly toward the opposite direction. So these are the second event. Next event, whatever the fibers get formed, जितने भी microtubules देखे they're like astral ray, spindle fiber, chromosomal fiber, interzonal fiber, all fibers they get disappear. Chalo, I'll write down all name together. It's better, I think. So first of all, we are talking about that is astral ray. Again, there is spindle fiber. Then interzonal fiber. Then chromosomal fiber, etc. Okay, etc. All these fiber they get disappear because now these are the end of the karyokinesis. There is a no requirement of the spindle fiber, so all they get disappear. Okay, third event. Now we are talking about reappearance. So you know very well during the prophase we already discussed there is nuclear membrane, even organelles like nucleolus, endoplasmic reticulum, Golgi complex, everything it is get dissolved, right? So again, whatever that disappearance means, whatever that organelle or the plasma membrane they get disappear, now again they reform. So next event we are talking about reformation or reappearance of plasma membrane, nuclear plasma membrane, exactly. Nuclear membrane, then nucleolus, then ER, endoplasmic reticulum, Golgi bodies or Golgi complex, etc. etc. So whatever they get disappear, all they form again. Okay, done. Done. And finally, two daughter nuclei get formed. Two daughter nuclei get formed, and such daughter nuclei simply we can represent in a monad condition. So quickly we are revising this part. We are talking about the telophase. Telophase nothing but that is end of karyokinesis, where chromosome they start the decondensation. Again, the daughter chromatid they reach up to their respective pole. Whatever the microtubules get formed or the spindle fiber they get formed, all these they get disappear. Means astral ray, spindle fiber, intrasonal fiber, chromosomal fiber, continuous, discontinuous, all kind of the fiber they start disappearing because they get degrade because the process of the karyokinesis is done. So there is no need of any spindle fiber. Again, during the late growth phase, nuclear membrane get disappear, nucleolus get disappear, endoplasmic reticulum they get disappear, Golgi bodies, everything they get disappear, and slowly, now all those organelles they get reappear during the telophase. And finally, there is a formation of the two daughter nuclei. Okay? So quickly we are taking the diagrammatic representation for the telophase. Chalo. So these are the outermost layer, that is plasma membrane. Plasma membrane. Inside the plasma membrane, now two nuclei get formed because common cytoplasm hai, because still we are talking about the karyokinesis, no cytokinesis. So inside the single cell, common cytoplasm is present, and within inside the common cytoplasm, two nuclei get formed. So one nucleus is present at the one pole, and the another nuclei they get formed toward the another pole. One nucleus, and now these are the another nuclei. Just draw equal size, okay? Because equational division, you know. One nucleus, and now these are the another nucleus. So simply we are talking about these are the cell where we are talking about these are the nuclei, single nuclei present. Okay, so basically these are the nuclear membrane, and these are also nuclear membrane. Inside the nuclear membrane, we are talking about there is a presence of the genetic material like this, which is present in a chromatin fiber. So obviously decondensation ho gaya, and due to decondensation, now whatever the chromosome is present, now that chromosome it is converted into chromatin fiber. They convert into thread-like structure. Okay. Now the dark body present hoga. The dark body nothing but we are talking about nucleolus, because during the end of the telophase, all that nucleolus they get formed. So this mustard color, simply we are talking about these are the organelles, that is nucleolus. Okay. Now toward the outer side, there is a presence of centrosome. Inside the centrosome, pair of centriole is present like this, which is present in a perpendicular manner, and obviously they can form a ray like aster ray. So these are the two poles. Obviously we are talking about these are the one cell having the common cytoplasm, but within the common cytoplasm there is a formation of the two nuclei, and whatever the entire structure, ye jo entire structure bana, or whatever the entire structure is get formed, this structure simply we are talking about these are the daughter nuclei. One daughter nuclei, these are the another daughter nuclei. Can you tell me this daughter nuclei is having which stage? Means during the telophase, which condition is present? Monad condition or the dyad condition? Quickly tell me, during the telophase, which condition is present, monad or dyad? Monad? It's monad. Okay. So definitely these are the represent the condition that is monad, and this another daughter nuclei also they represent the condition that is monad. That's it. At the end of telophase, whatever the initial diagram, obviously when we start from the prophase, per cell only single nuclei is present. But now at the end of the telophase, we obtain exactly two set of nuclei. All the organelles means whatever the condition means common cytoplasm, but all the material they get double and two nuclei get formed, and that both the nuclei they represent the monad condition. Got it? Chalo, quickly complete the diagram. Two daughter chromatin are the collectively called as the daughter chromosome. Yes. Collectively they are called as the daughter chromosome. Inside the daughter nuclei, daughter chromosome is present. Whatever the genetic material which is present at two end or towards the two pole, it is represent the daughter chromatin. Okay, but you have to remember, okay, whenever we are talking about the part that is daughter, you have to remember two respective pole of the cell, two respective end of the cell. You have to remember those points. Chalo. So now all these are the information with respect to karyokinesis. Initial phase that is prophase. Now cell entered into metaphase. Now third one that is anaphase, and finally there is a telophase. Done. Now we are talking about last part of the mitosis, that is cytokinesis, cytoplasmic division. Cytokinesis, that is cytoplasmic division. Okay. You know very well, karyokinesis is followed by the cytokinesis. Initially, we have to divide all nuclei, and once nucleides get divided into two daughter nuclei, now cell proceed to cytoplasmic division. So now, what happens? Such type of the cytoplasmic division, it is get vary in plant as well as the animal. So basically, it is a two different way. In case of the animal, cytokinesis is quite different, while in case of the plant, cytokinesis is get different. When we are talking about the animal, during the animal condition, there is a formation of the cleavage furrow. Cleavage furrow get formed, right? Cleavage furrow. And cleavage furrow, nothing but in other language that is also called as invagination of plasma membrane. Cleavage furrow, simply it is called as the invagination of the plasma membrane. We'll see this part during the diagrammatic representation, don't worry. So remember, during the animal cytokinesis is quite different with respect to plant cytokinesis. So in case of the animal cytokinesis, simply plasma membrane, they are showing invagination, right? And such type of this process simply is called as the cleavage furrow. And when we are talking about the animal cleavage or the animal cytokinesis process, now there is a simply movement of plasma membrane from periphery to center. So invagination of the plasma membrane means what? That is, it is also called as the movement of plasma membrane from periphery to, periphery means toward the outer side, from periphery to center. Okay, periphery to center. And such type of this condition, it's very much common, it is very much present in case of animal cytokinesis process. When you compare this point with plant cytokinesis, in case of the plant cytokinesis, now there is a formation of cell plate or also called as the pharmoplast. There is formation of cell plate or such cell plate also called as pharmoplast. Pharmoplast. Underline this word. Cleavage furrow, please underline. Cell plate and the pharmoplast, please underline. We'll discuss this part during the diagrammatic representation. Okay? So, cell during the plant cytokinesis, obviously there is a formation of the cell plate, and that cell plate also called as the pharmoplast. Now, when we are talking about such kind of this division, obviously we are talking about that cell plate they form at the center. So, whatever we are talking about, cell plate. So, this cell plate form at center of the cell. They form at the center of the cell, and now such plate they are showing elongation from center to periphery. Cell plate simply they are showing elongation from center to periphery. Division pattern different. When we are talking about the animal cytokinesis, obviously we are talking about there is a formation of the cleavage furrow, and in case of this cleavage furrow, obviously there is a invagination of the plasma membrane. When you compare this point with the plant cytokinesis, during the plant cytokinesis there is a formation of the cell plate also called as the pharmoplast, and simply we are talking about that is cells, these are showing movement from center to periphery. Clear? Yeah. This point is clear, everyone? Any doubt from today's class? Any doubt from today's class? Hello? Any doubt from today's class?
No, ma'am.
No, na? ठीक है चलो. So with this we end today's session, and next class will continue with the diagrammatic representation for the cleavage furrow which is occur in animal and cell plate formation in case of the plant, and then we'll talk about the further process, significance of the mitosis, some drugs who act as a activator for the mitosis and who inhibit the mitosis process, and after that we'll start with the second parameter of this chapter, that is meiosis, reductional division. Done?